reactions that are happening in living systems, right? Okay. So what is the importance? Its importance is in all the different fields. That means in medicine, in pathology, in medicine, biochemistry, it's a guide to drug discovery and its application. In pathology, that means when you go to a pathological lab, if you look at the report, you will see that there is a sign of a biochemist. So there are different tests, biochemical tests, which are performed to diagnose the disease state. Then it is used in agriculture. Now the microorganisms that are present in the soil, there are microorganisms everywhere, they are present in soil, in plants, even the plants itself and certain animals, they help to develop the organic manure. That means they increase the fertility of the soil, and the biochemical reactions in this system are actually helping them to increase the fertility. Pharmacy, now how drugs, when you are administered a drug, then how it is metabolized in your system. The metabolism part is solely driven by the biochemical reactions. And food and nutrition, that means nutritive value and quality of the food is also assessed by assessing these biochemical reactions. Now quality of food, one such experiment we are performing, that is we are checking the quality of raw milk. Now the biochemical reactions, they are basically of 4 types, that is the neutralization reaction, that is acid-base reaction, redox reaction or the oxidation, one is oxidized and another is reduced. Condensation reaction. Now condensation reaction is basically when a macromolecule is being synthesized, 2 monomers are joined together with the release of a water molecule. So macromolecule synthesis involves several condensation reactions, and hydrolysis where a macromolecule is broken down to evolve energy which is utilized for the living system functioning. Now all these biochemical reactions are facilitated by biocatalysts. Catalysts you all know, and biocatalysts, what are biocatalysts? These are enzymes. Now the enzymes not necessarily means proteins. Enzymes can be proteins. Majority of the enzymes that we come across that are present in our body, they are proteinaceous in nature, but enzymes can also be RNA. Okay? So enzymes, when we talk of enzymes, it's both protein and RNA. Now, if you look at this pathway, this is actually a series of biochemical reactions that are going on in our cells, in any living organism, in a bacterial cell. Now, this pathway is a pathway which shows the metabolism of a carbohydrate molecule, which is being performed here, right? Because we have added sugar and yeast. And here you can see here, there is some part is, this part is degradation or breakdown of the molecule, and these parts, that is nucleotide synthesis, lipid synthesis, some of the components or metabolites are being used for synthesizing lipid and nucleotides. So we won't go into the detail of these pathways, we will just concentrate on the basics. Now this all know, right? This is essentially what we are looking at. It is the fermentation by yeast, where this is the glucose. Now sugar, what is sugar? What is the sugar that you have added? It's not glucose, right? It is sucrose. So what is there in sucrose? Yeah, it's actually a glucose molecule and a fructose molecule. So it will be broken down to glucose, and fructose will be converted to glucose, and ultimately there will be breakdown to 3 carbon, 2 carbon, and it will lead to generation of ethyl alcohol, which is used in wine production, and carbon dioxide, which is used for the synthesis of bread and cakes. And this is the organism that you have added, right? Now here you can see there is this reduction, oxidation reduction, there is the breakdown or hydrolysis, and this is the energy production, because ATP is the energy currency of the cell. Okay, now how biochemical reactions are used for bringing at the dairy farm, right? So at the reception, these raw milk samples are checked. And the test is a very simple test that you have performed. This very test is being performed at the reception to check whether the raw milk is of good quality or bad quality, or whether it can be accepted. So, what is milk? Milk, you can see majority of its water, and these are the different percentage of carbohydrate, lipids, proteins and minerals. So it is a complete food for us as well as for the microorganisms. Now the microorganisms, there are certain microorganisms which are the normal microorganisms that are present in the milk, and these are the few to name. However, there are certain pathogenic microorganisms that, because it's a very good source of food, so this pathogenic microorganisms, if they are contaminated somehow, if they enter the milk, then they can easily propagate inside the milk. So the basis of this reaction, or the aim of this reaction, is to check the presence of the microorganisms. Now, the microbiological quality of the milk is very important. Microbiological quality means we are not talking about the normal microflora. We are talking about the pathogenic microorganisms, right? And from where these pathogenic microorganisms can enter the milk, these are the possible causes of entry, right? That means poor milking methods, long duration of transportation, inadequate cleaning, poor cooling, and mastitis, which is an infection that occurs in the cow, right? Okay. Now, what is this reduction test? Methylene blue is a dye, okay? And the milk, the microorganisms, the content of the microorganisms in the milk is, or will be, very high if it has the pathogenic microorganisms in it, okay? So as the number of microorganisms increases, the dye which you have added, that will actually indicate the presence of more number of microorganisms. How? Because the microorganisms, for their metabolic processes, they need oxygen. They will use up the oxygen. So the oxidizing environment is gone, and the methylene blue dye will be reduced, and in its reduced form it is colorless. So the speed of reduction of methylene blue is directly proportional to the number of bacteria present in the milk sample. So the normal microflora present cannot decolorize the milk, that means the methylene blue that you have added in the milk, but if there is contamination and the number is more, that can decolorize the milk, the methylene blue. So this is basically the structure of the methylene blue here. This is blue, this is the oxidized form, and this is the leucomethylene blue, which is colorless. Okay, so if you look at these tubes now, you can see here, this is the A milk, right? Initially it was blue, now gradually it is turning white. Obviously it needs some time. Whereas if you look at this tube, okay, this is the control milk. We haven't added anything here. This is the methylene blue added milk, which has not been yet decolorized. And if you compare the color gradient, right? So you can see this milk, it has decolorized, but whereas this, it hasn't. Okay. And what about this? Right? So you have your yeast-air balloon. Okay? So what is there in it? Carbon dioxide, right? So it's a very simple experiment which you can perform even at your home. Okay? And if you have younger brothers and sisters, they will just love it. Already have some microorganisms, right? Normal microflora. Those are not harmful. So obviously if you add this methylene blue and if you keep on the milk for a very long time, ultimately it will be decolorized. But that will be due to normal microflora. But if it is decolorized faster, that means there is cross-contamination of the pathogenic microorganisms. So this is the decolorization time and the result. So if it is decolorized from 30 minutes to 2 hours, it's a poor quality, 2 to 6 fair quality, 6 to 8 good, and over 8 hours excellent. So actually these two should be accepted and these two should be rejected from the reception counter, right? Okay. Okay, now we move on to perform another experiment. Again, I will ask for some volunteers. From the back. Yeah, 2 volunteers will be fine. Okay, I think all of you know hydrogen peroxide. What happens if it is left alone? H2, water and oxygen, right? And what is the catalyst for this? If it is left alone, it will decompose, but it can be done faster with the use of a catalyst, chemical catalyst. Okay, we will just see. So this is hydrogen peroxide, right? So come on and I will just read it for you. Now add 4 drops of this liquid soap. 2 or 4. Okay, fine. Now you add, this is potassium ion. Okay, just add a little. Just a little. So what is happening? Liberation of O2. And why this O? Because we have added the soap, right? And oxygen is creating this foam. And so what is here? We have added potassium ion, or you have added. So what is that?
Potassium ion is a catalyst.
It's a catalyst, right? So it's a chemical catalyst. Okay, thank you. Go back to this. So this is just the catalytic decomposition of hydrogen peroxide, right? Now this same decomposition takes place in our body. Okay? So do our body produce hydrogen peroxide? Yes, we do. All cells do produce hydrogen peroxide, right? And we also use hydrogen peroxide. Where? In day-to-day life. May not in day-to-day. Some may use, some may not. It is actually a disinfectant. Okay? 3%, 1.5% to 3% solution of hydrogen peroxide is a disinfectant. Okay? So this hydrogen peroxide, it is why it is a disinfectant? Because it generates hydroxyl radicals, which it will affect the site of infection. Okay? When you add hydrogen peroxide to the site of infection topically, it will remove the cells because it will damage the DNA, and that will result to cell death. So it is used as a disinfectant. Now in our body or even inside the cells, hydrogen peroxide is produced. How or when? It is the aerobic bacteria. Aerobic means, I think you know what is aerobic bacteria, right? Those which can utilize oxygen or grow in presence of oxygen. Facultative aerobic also have also produce hydrogen peroxide, and even in our cells, that is in liver, majority of detoxification processes takes place in the liver cells, right? There a lot of hydrogen peroxide is generated, and hydrogen peroxide, mind you, it's toxic, right? That is why we use it as disinfectant. So hydrogen peroxide, when produced inside the cell, should be rapidly degraded. For that, there are biocatalysts. What are they? These are the catalase enzyme and the peroxidase enzyme. Their function is more or less same. They decompose this hydrogen peroxide, right? And they produce water and oxygen. Peroxidases are present in the anaerobes, and catalase is present in the aerobic organisms as well as in our body. Okay? Now, this is the structure of the catalase, which is protein. This is the crystal structure of catalase. So, the last talk you heard a lot about the crystals, right? So, this is the crystal structure of a protein molecule. Now, this is just the representation of the chemical reaction that is going on. So, this is the chemical reaction that you have just performed, and this same reaction takes place in the living system with the help of catalase and peroxidase. So, we have seen the chemical decomposition of catalase, and now we will see the decomposition of catalase in the living system, right? So, for that, actually, okay, some of you can, if some 2 or 3 people can come forward. Okay, this is a bacterial culture, a culture of E. coli, Escherichia coli. I think you have heard the name, right? So, this is a culture of Escherichia coli, and so I will just add hydrogen peroxide to these tubes. Take one glass straw, each of you. I think it will be better if we do this experiment there. Okay. So one by one, take some culture from this, just with the glass straw. Scoop out some culture.
Please, show inside.
Okay, do one way. And you just dip it here. You can hold this here. What you see? Bubbles are coming out, right? Right? So what does that indicate?
Decomposition of
H2O2, right? And who is doing that?
the bacteria.
The bacterial cells. So it is the presence of catalase or peroxidase. This is a E. coli cell, so it is aerobic. So it's a presence of catalase in the bacterial cells that is being demonstrated, right? So you also can do that same thing. So you can see a lot of oxygen, but tube. Just this is a so this will also eventually, this is just hydrogen peroxide. This will also eventually be decomposed in water and oxygen, but since it is not catalytic, right? It's slow, so we cannot visualize that. So just take the glass straw. Then you can see the effervescence coming out, the culture. Try to keep it on the glass straw. Yeah. Just hold it for a moment. Yeah. Here it is coming out.